We see extraordinarily diverse adaptations in the living world, bacteria that can live in the most challenging environments, complex organisms that develop reliably from a single egg, and the elaborate cultures of ants and humans. These have all evolved through the simple process of natural selection, acting on random variation. We try to understand this evolutionary process, focusing on populations that are distributed through space and selection that acts on large numbers of interacting genes. To understand how species adapt to their environments and how they split into new species, we need to know the effects of spatial subdivision and the flow of genes from place to place. Conversely, spatial patterns can tell us about the strengths of evolutionary processes that are hard to measure directly. For example, the strength of the selection that maintains sharp boundaries between populations. Most of the traits that we're interested in depend on large numbers of genes, and we cannot hope to understand all the myriad details of their interactions. We try to understand constraints on the evolution of gene networks, and to develop simple models for the evolution of complex traits that do not depend on all the underlying details. We try to make sense of the flood of sequence data by finding robust ways to infer population history and to detect selection from patterns of genetic diversity. This work demands a combination of mathematical theory, analysis of genomic data, and fieldwork on natural populations. And so this work is exceptionally diverse, including people with backgrounds from mathematics and physics to genetics and ecology. This diversity is challenging, but it's also fun.
Some of us in the group use snapdragons as a model system to understand evolutionary processes more generally. The genetics of flower color in snapdragons has been worked out for a long time, but we want to study natural selection on flower color in the real world. So I work to understand why flower color experiences natural selection in snapdragons. Does flower color affect plant-insect interactions like pollination or seed predation? Does flower color matter for dealing with stress like drought? This kind of research sits right at the intersection of ecology and evolution. So I collect field data, I do greenhouse experiments, and of course I do lots of fun statistics to deal with complicated data from the real world.
But it's not enough to just study the evolution of traits. We also need to study the evolution of the genes that underlie them. In the past, studies of genetic variation were limited to very short gene sequences, meaning that we never had enough data to really test long-standing evolutionary theories. In the Barton lab, we're using the latest whole genome sequencing methods to study and understand how genetic variation is shaped both across space and time by processes like natural selection. And we really study on two major model systems in the lab at the moment. The first is the Snapdragon system, and also we study Litorina snails. And so although we do focus on these model systems, the work's really motivated by more fundamental questions in evolutionary biology.
Ultimately, our understanding of evolution is only as accurate as the theoretical models that we use to describe it. For my PhD, I'm simulating realistic spatial models of evolution. I use the long-term spatial and genetic data we have collected on snapdragons to inform my simulations, which helps bridge the gap between theory and data. In turn, my simulations will inform the development of more realistic evolutionary theory. My research will also help with new methods for inferring signatures of population structure from spatial genetic data in the present. This detailed understanding will have direct implications in conservation biology, population management studies, and so on.
Not everybody in the group works on snapdragons. I'm actually studying why and how females and males look so different, which is a relevant question because they share the same genome. So evolution needs to come up with clever ways to decouple sex-specific gene expression from a shared set of genes. And this is actually also related to other questions, like for example, how do tissues differentiate in an individual? Like for example, cells in the epithelium or in our eye look completely different, but they come from the same genome. So I'm basically studying the reasons why sexes look so different and how this difference evolve over time.
Working in the Barton group is not just about research. Love of nature is something that connects us. We like to spend time outdoors, hiking in the mountains, swimming, exploring natural wonders, and doing active sports together. Our field season is also a very special time where we can spend time in Spain together and enjoy Spanish culture. People in the group are from all over, so we also learn about Austrian traditions together. We will wander around Christmas markets, visit local wineries, and of course explore Vienna. I truly believe that our passion for science unites us and serves as a foundation for many great friendships.